Hi everyone, my name is Santacruz. I'm a PhD student at National Autonomous University of Mexico. I'm really happy to share my research with you. I developed this project with my two advisors, Dr. Marta Barluenga and Dr. Gerardo Pérez-Ponce de León. The title of my talk is Host-Parasite Interactions in the Recent Adaptive Radiations of Midas cichlid fish in Neotropical crater lakes. Remote and isolated environments, such as the small oceanic islands, without apparent geographic barriers for genetic flow between populations, are an ideal setting to look for sympatric speciation. Small isolated lakes are like islands. Some species colonize and form unique communities. Some lineages are able to diverge and others not, and these mechanisms include all forms of life, both hosts and parasites. In a classic scenario of sympatric speciation, once colonization occurs, it begins with a population with random reproduction and constant gene flow. At this point, the population has a normal distribution of the phenotypic variation. These populations could adapt or prefer different environments and have different diets, so if the mating choice is linked to the habitat preference, this automatically generates reproductive isolation. Therefore, natural and sexual selection are in action. However, if these populations are under divergent selection, we can add one more factor. These populations could be exposed to different parasites. Parasites are able to exert a strong selective pressure on their host. This could generate local adaptations, for example, fish that look, behave, or smell different. Then, females could choose males that are more resistant to the local parasites, promoting reproductive isolation. So, this parasite-mediated selection could contribute to species divergence. This led us to our main approach, parasites as drivers of divergence in their host. This will have to meet two assumptions, which represent our hypothesis. First, parasite-mediated selection should differ within or between host populations in terms of parasite abundance or community composition. Two, the direction of parasite-mediated selection between host populations should be stable over time. This question has been addressed in model organisms such as the stickleback Gasterosteus aculeatus, a minnow that inhabits the northern hemisphere and is one of the most extraordinary examples of evolutionary parallelisms. In this fish, populations in contrasting environments have different parasite communities. Furthermore, there are specific alleles of the immune system to respond to particular parasites. These alleles can change rapidly depending on the parasite to which one is exposed, and females choose the male who responds best to the local parasites, which leads to rapid adaptations. Other example is the subspecies of birds where the sexual signals are associated with the most costly local parasite. Another example is in the Arctic char, where the coloration is associated with the parasite's transmission strategy. So why study this approach in the Midas cichlid? One of the reasons is the geographical setting where the Midas cichlid species complex is evolving. This fish inhabits lakes that lies on one of the most volcanically active areas on the planet. The system is within the territory of Nicaragua. It is made of two large tectonic lakes, Managua and Nicaragua, plus a chain of adjacent small crater lakes which are completely isolated. Another reason, and obviously a great pleasure to our study, is that they are really beautiful environments. As you can see in these photos, each crater lake is a caldera of an inactive volcano that is filled with water and in some way also filled with life. Here are the lakes. Each of the crater lakes were colonized independently by populations derived from the Great Lakes, Managua and Nicaragua, along a very recent temporal gradient, only between 4,700 and 800 years ago, which include only a few fish generations. Therefore, this colonization has followed a continent island model. In some lakes, the Midas cichlid species have diverged in sympatry, while in others not. In the lakes Apoyo and Hiloa, the Midas cichlid have formed adaptive radiations. In other words, the complex includes homogeneous populations to flocks of many endemic species, capturing the so-called continuum of speciation. The adaptive radiations in Lake Apoyo and Lake Hiloa are among the few accepted examples of sympatric speciation, constituting a textbook example. In several lakes, there are evidence of parallelism, but at least in two lakes it is very clear, Lake Hiloa and Lake Apoyo, deep and transparent lakes that have converged in replicated phenotypes according to the habitat preference. They converge into bedding forms, the well in the shallow or deep littoral zone, and phenotypes the well in open waters with a lignetic body shape. Other traits related to divergent selection are thin and thick leaves size, ecomorphotypes, and color variations known as dark or gold. With this idea of testing the hypothesis of parasite-mediated selection, we set several goals. Today, I'm going to talk about these three. First one, it's identify the parasite fauna of the Midas cichlid fish. Two, characterize the parasite community's assemblage. And three, study the phylogeographic patterns of the parasite species. In order to carry out this, we screened about 900 fish over 3 sample seasons, with a major effort in the Midas cichlid, but also in other cichlids and other fish families that coexist in the lakes to determine patterns unique to the Midas cichlid. As a study area, we include 5 crater lakes and the 2 large lakes. So, we have populations derived from both Great Lakes and more ancient and young populations. We made the sampling during the reproductive season, time where it's possible to observe the parental care, as well as pairs of conspecific with the same coloration. We collect fish from different environments within the lakes, including deep areas. Then we score all the macroparasites on the same sampling day, and we store and count absolutely all the parasites. So far, we recovered thousands of parasites, so we have found an extraordinary taxonomic diversity, including very distant taxonomic groups and all kinds of life cycles, parasites that mature in the fish or that use the fish as an intermediate host, or others that complete their life cycle in or off of the water, for example, in birds or mammals. That is 42 species, including 9 groups of parasites, mainly helminths, involving the description of new species. Within this species, a subset of 18 parasites infects non-exclusively the Midas cichlid. As expected in a continent island model, there is a high diversity of parasites in the Great Lakes, Managua and Nicaragua, but in most lakes, all groups of parasites are present just in different proportions. However, it's extremely interesting that in very small lakes, there is a high diversity of parasites and high levels of endemins, for example here in Asososca, León. Within this universe of parasites, a group of 8 parasites infects the Midas cichlid throughout all the lakes and in most of the species that we name the core parasite fauna. Thus, the Midas cichlid is interacting with a group of endo and ectoparasites. Therefore, the core parasite fauna is widely distributed through all the lakes. All the species inhabit the Great Lakes, and in some crater lakes, there is a recurrent absence of some parasites, coinciding with the most recent Midas cichlid populations. This means that these species tend to occur more frequently than expected. So, if you have one of the species, it is likely to find the other. This may involve similar filters associated with colonization and their presence within the lakes, while for the rest of the species, most of the occurrences are random. In this ordination analysis and in MDS, each point corresponds to the parasite community of a single individual, while in the graphite lake, it corresponds to the entire host population of the same ecomorphotype. The more similar the parasite community are, the closer the points are to each other. In the graphite lake, the fish are ordered according to the habitat. We see different patterns throughout each crater lake. So, if we look to the general patterns in the whole system, the result displays that each lake has a unique parasite assemblage. This will be the outcome of an intralacustrine radiation. That is, the Midas cichlid may have colonized with a subset of their parasite fauna from continent to island, and then the parasite communities were assembled in concordance with the divergence of their hosts. In addition, they resemble the patterns of colonization and radiation that have occurred in the Midas cichlid. The parasite communities are significantly different with no year fed, which meets one of our hypotheses of temporal stability of the parasite communities. The same parasite communities, according to the color of the fish, indicate that gold versus dark have a totally different pattern, independently of lake origin. The Gulf phenotype that tends to low frequencies in all the lake system, besides, is an ecomorphotype associated with rocky environments and turbid or deep waters. We will take this small lake as an example, where only one fish species lives, but which has phenotypes that tend to mate according to coloration, also occupy different environments. We found that there are significant differences in the infection patterns, with the Gulf phenotype infected by less parasites. Let's consider another phenotypic trait, such as the lip size. In a recent lake like Apoyeque, just 900 years, there are thin and thick lip phenotypes that are not yet fully differentiated, but made according to lip size. In accordance with our hypothesis, if there is a divergent selection mediated by parasites that contributes to speciation, we should observe a very visible effect of the parasites. In agreement, we found significant differences in the infection patterns. Furthermore, if the new phenotype is moving to a new environment, we will expect that in the first stage of divergence, the new colonizer will have less parasites. If we look at this same trait of the lip size in lakes where the species are completely differentiated, we also observe differences in the infection, although in different directions, but parallel in both lakes. I want to point out that there are differences in parasites at different points of the species divergence. My next question is whether there are also differences in parasites among sympatric species, and the answer is yes. If we do the same comparison between parallel phenotypes from different lakes, there are also differences among sympatric species, but the infection patterns doesn't look parallel. As a first point, not all species of parasites are shared between both lakes, in addition to the historical contingencies involved in the assembly. However, if we take a closer look at the transmission strategies of the parasites, we observe parallel patterns. The lignetic phenotype has more nematodes according to its predatory behavior and its position at the top of the trophic chain. Another parasite with a totally different transmission strategy, which depends on the physical contact rate between its hosts, we found that in both lakes, the fish that inhabit the shallow zone of the column water has more parasites, and precisely these fish tend to have a higher density associated with the littoral zone, while fish in the lignetic zone has less physical contact. So far, we have addressed patterns across all the lake system and within host species in each lake. So the next level is what happens within each host. The fish represents all the environment for the parasite. So, as in any community, dynamics of cooperation or competition can occur, depending even on the specific niche or habit that each parasite occupies within its host. I found that different interactions occur in each fish. Most of them are positive, only one was negative, and they are not replicated between the parallel phenotypes between lakes. In other words, the interactions can be influenced by the traits of the host or the life history of each parasite. Positive correlation should be associated with similar requirements or host susceptibility to a major diversity of parasites, or likewise, infection with one species could facilitate the presence of another. Anyway, there are many non-mutually exclusive mechanisms. In the lake system, the Midas cichlid is the only group that has radiated, but other cichlids also present a wide distribution. So, here I show you the general patterns through all the lakes in the Midas cichlid against a lineage that didn't radiate. In the lower bar, here, the color represents lake in which the fish lives and the circles the relative abundance of a particular species of parasite. The same species of parasites are shared with the lineage that didn't radiate. Fish with radiation do not replicate the infection pattern of any cichlid population. The fish that didn't radiate has heterogeneous parasite communities among lakes. Some parasites have similar abundance in all the hosts, independently of the lake. And finally, if we have considered all these patterns in contrast with many more fish species, and the fish most phylogenetically distant share less and therefore share less parasites, and therefore its parasite communities are more different. We have studied in great detail the genetic patterns of the most common parasites, the core parasite fauna, not only through the legs, but also the genetic variation within each host. We found that each parasite has a different evolutionary history, independent of its host. But interestingly, some groups display high genetic structure, within characterized with more fish species. For example, with unique haplotypes in Lake Apoyo and Lake Hiloa. Here and here and here. One particular example is this species that I described, Procamelanus barlowi, which is present in all the lakes except one. It displays unique haplotypes in Lake Apoyo, different from the rest of the lakes, a pattern that could imply sympatric speciation. This parasite also has different lineages that coexist within the same host, implying a possible intra-host speciation, or as I mentioned, speciation with gene flow in a parasite. Finally, with this study, we have characterized the diversity of parasites in the Midas cichlid and other coexisting fish. We found parasite communities differ among host species in sympatric and allopatric and are stable over time, forming unique assemblages of parasites in each lake. We found signs of parallelism associated with transmission strategy of the parasite. We found unique interactions among parasites in each host community. Each parasite has a unique evolutionary history in the lake system, and one of our most important conclusions is that parasites could be a factor promoting divergence of their host from the first stage of incipient divergence. Our main next step is to associate the infection patterns of the parasites with the variation in the immune system of the Midas cichlid. Finally, this work is not possible without the support of many people in the field and in the laboratory. I really want to thank you for your attention and invite you to contact me with any question. Thanks.